This is a production of Cornell University.
I think the previous speakers, they gave a nice overview of how animal science and plant science have worked together, and some of the concepts from one field were used in another field. So this cross disciplinary learning was really important in driving the plant breeding and as well as animal breeding in the past. I'll give you some personal example of how this cross-disciplinary learning have helped us answer some of the complex questions in field phenotyping. This is an example from our lab. So we started in 2014 with the UAV phenotyping project. I joined Jesse's program in genetics in 2014. And right about that time, we were starting to explore UAV phenotyping and how we can use this phenotyping in breeding. So we got involved in that project. We had engineering people from engineering department. Kevin Wong, he was a postdoc in bioag engineering. Dale Shinstock, he was a mechanical engineer. So we started a UAV testing program. So we started flying UAVs, testing new platforms to see how which of the platform fits well with our goals. We did a lot of testing and crashing too, but good thing is it was a cheap UAV, so we could afford. So the arms and legs were like very cheap, so we could replace them. So a lot of learning in 2014, and then 2015 we had people from geography and bioinformatics joining in, and really helped us move ahead with the learning some other things. For example, we narrowed down to sensors, what kind of sensors we can use, and so we picked 1 or 2 sensors that were really working well with our program, and we made a lot of progress in analyzing this data. 2016, we had a aviation engineer joining our team, and he really helped us collect a lot of data in the field and really ease my work, because I was doing a lot of flights and the genetics which is my core field, I totally was behind. So he really helped me. So then I started focusing on the genetics and then using some of these data sets that we generated using these new platforms and sensors. So this is really to highlight how cross-disciplinary teams can really bring in new learning and how we can use these approaches to answer some of the complex questions. So ultimately, like this cross-disciplinary approach and all these things. So our main goal in plant sciences and even animal sciences is to connect genome to phenome. So, but it's a very crucial link if you want to improve the crop productivity. But we also have to address this field phenotyping bottleneck at the same time, and this high throughput phenotyping can really help us to understand this genotype to phenotype link. This HTP can allow us to rapidly and accurately measure traits, and especially in the field, which is really a bottleneck. So we can test a large number of plots in the field, which can really help us study these complex traits. Some of the traits which are really hard to measure, and I'll give you an example today of lodging. So a little bit of what we are doing in the lab, and some of the studies that came out from our lab. We had a few studies showing how this new HTP tools can be used in large breeding nurseries. Really, one of the cool thing about UAV phenotyping is that we can view this trait in a dynamic way, because we can measure these phenotyping. We can take these measurements, plant measurements, multiple times during the season, and that allows us to look at the trait in a dynamic way. And we can really see how the things change over time during the season. So rather than just looking at a single point, we can think of it as a dynamic trait. And this is an example. So UAS for lodging assessment in wheat. This is what I'll be talking about mostly. So lodging, this is how it looks from the sky with the drone. You can see that there are lodge plots, and there are plots that are just unaffected by lodging. So we thought initially, like when we saw these images, I was collecting data regularly for height and NDVI and other things, and then I saw these images, and then Jesse and I we talked, and we thought, can we quantify this lodging using these image-based approaches? So just a little bit of background. Lodging is a permanent displacement of plant stem from its upright position. It can have a severe impact on yield and quality in wheat. But lodging assessment in field is very laborious, and it's subjective in nature. So this limits our population size for genetic studies. So this limits how many plots we can study in the field. So that was really the driving force for us to use high throughput phenotyping, so that we can really address this issue. So the study objective was really to develop a US-based lodging assessment approach. So really to see how we can use these image-based approaches to quantify lodging at the field scale, and then also to come up with the proof of concept, how we can use these phenotypes, the image-based assessments, to study the genetic architecture of lodging in wheat. So our field setup was, so these are yield trials, CIMMYT yield trials in India. We had this experiment going on in the field. I was already taking data, as I mentioned. So what I did was I basically picked the two dates, pre-lodging date and post-lodging date, and generated a digital elevation model from these dates in both years. So we had close to 600 entries in both years. So, and also at the same time, I collected ground truth data just to compare how these image-based approaches that I'm going to test on do compared to the visual measurements. So the approach that I used was based on digital elevation models. I took a pre-lodging digital elevation model, subtracted it from a post-lodging digital elevation model. So really to get a differential DEM, which is a change in elevation over time. So these two time points. And then use this differential DEM in each plot to really come up with two different indices. One is a very simple index, which I call differential lodging mean, or DL mean, or digital lodging mean. So this is basically just taking the average of all the differential points within a plot. So that's the first index. Second index is a DL mixture, I call. This is based on a more informed normal mixture distribution-based index. So a little bit of how the DL mix is calculated. So basically we have distribution. This is an example from a single plot. So we have two normal distributions. One corresponds to the non-lodged part, and another one is a lodged part of the plot. So we basically estimated the parameters of each distribution, and came up with a lodging index multiplying these two parameters from the lodged part of the distribution. So now I have two digital lodging indices and three ground truth measurements. So I took lodging incidence, which is a severity 0 to 100%. Lodging, sorry, this is the incidence, and then severity is on a scale of 0 to 10. How much of the plot is the first one is how much of the plot area is lodged. Second one is how severe is the lodging on the scale of 0 to 10. So then I also combined lodging index and lodging severity to see how this compares. So we explored phenotypic relationships on the diagonal. You can see the first three are the visual traits, visual assessments of lodging. The last two are the DL mean and DL mix. And in the parentheses, these are the broad sense heritabilities. So this is 2016. We had comparable heritabilities, broad sense heritabilities, and very high correlations among visual and the digital assessments of lodging. So our image-based assessments were working at this point. I also looked into the genetic correlations. So genetic correlations were also very high in both years, above 0.9, so which was really good. So we had evidence that what we are measuring is actually is lodging, and we're measuring the same phenotypic variation. So once we had these validation results, we wanted to look into the genetic architecture and see how we can use some of these approaches in understanding the genetic architecture of lodging. So in 2016, this is a Manhattan plot from 2016, first year. There were no significant marker trait associations. In 2017, however, there was a peak at chromosome 2A. This peak corresponded to translocation segment, which is a translocation segment from Agilops ventricosa, which is a wild ancestor of wheat. This 2NS segment is involved in a lot of, so it has been extensively used in breeding programs historically, because it has a lot of disease resistance genes. And this peak was really consistent across all measures of lodging. So that was a good thing to see. But an interesting thing was that the stop markers only explained up to 2% of the variation. So the variation was very low. The variation explained by these markers was really low, suggesting that this could be a really complex trait, and trait with a diffuse architecture with large number of small effect loci. So to explore that hypothesis of diffuse architecture, we generated a Kfold genomic predictions. So we wanted to look how if we can explain some of this unexplained variation through whole genome prediction models. So here is a comparison in 2 years, 2016 and 2017. There's a prediction accuracy, and then prediction accuracy scaled to the square root of heritability to show how the proportion of heritable genetic variation that is explained by the prediction accuracies. So we were able to explain up to 53% of the heritable genetic variation, which was really nice to see. And we also tested it across environments. So cross environment prediction accuracies were also high. Here we are comparing the prediction accuracies, phenotypic correlations, and genetic correlations. So the data from Ludhiana location, where I collected UAV drone data, I used as a training set to predict the lodging at another location in Pakistan. So they had lodging index values that they collected in the field. So I was able to compare it across environments also, and the results were also encouraging across environments. So we were so this was another validation and another proof of how we can use some of these UAV-based traits to to look into the genetics of complex traits. And then we were also wondering if we can establish a relationship of lodging with the agronomic traits. One of the interesting things we saw was with the correlation of lodging with grain yield. So in 2016, so this is a result from 2016. Correlation was negative. So the relationship is opposite. So you have higher lodging, lower yields. 2017, however, was different. The relationship got reversed. So you have lines that are high yielding, they are more prone to lodging. So either of these cases can happen in the field, right? So some years you can have lodging at a stage where lodging happens earlier, and then it leads to yield losses. In other cases, you have lodging when crop is already headed and it's heavy, and then so it makes the high yielding plants more lodging prone. So that was interesting to see. So just quickly, the take home points. I don't know how I'm doing time. Okay. So hopefully I've provided some evidence of the scalable assessment of lodging on thousands of field plots. So we were able to do that in the field. This field-based high throughput phenotyping can be used to study complex traits in plants. We have that, so not only just study, we have other examples from other studies where we can use some of these new tools to study the complex traits in field. So genomic predictions can adequately explain the heritable genetic variation for complex traits with complex genetic architecture. Digital lodging can be integrated with the current breeding and prediction pipelines. A lot of breeding programs have started using this UAV-based approaches. So these approaches, so having a new trait, from this study, they can easily use this, easily incorporate these new tools into their breeding programs. UAV programs can really benefit from the predictions and other things. So finally, thanks to my advisor, Dr. Jesse Poland at Kansas State University. We have a great team at Poland Lab. Then we have great collaborators at Borlaug Institute for South Asia in India. CIMMYT Mexico has been a good big support. Ravi Singh's program on wheat. We have CIMMYT Pakistan, our collaborator Mohammed Imtiaz. So they were a great asset. So, and our funding agencies. Thank you.
Thank you. That was terrific. We can open it up to questions now.
Yeah, how far off of the center line of those plots did you have to fly in order to get that height measurement? When you're flying directly over, you can't see the height, right?
No, yeah. So when we are, when we plan our flights, we have a 70–80% overlap between the images. So we make sure that we have this 70–80% side and forward overlap. So basically, using this overlap, we can create a 3D model. So we have 3D points. So that's how we could get it. So this height was extracted using the z value. So you have x, y, and then with the 3D model, you can have a z value. And the z value is your height. So basically, you can do height, and lodging is an extension of height.
So in terms of the, did it lodge because it was super weak and thus that'll be low yielding, or because it was too powerful for its own good?
Right.
And I mean, so how many time points did you do the flyovers and have the digital elevation models for? Like how many times a year did you do a flyover that you could get the digital elevation models from?
Just once.
Okay, so if you did it at like 3 time points, you would probably.
Yeah, actually I have more time points. So you mean like different days?
Yeah, if you did it once early in the season and late in the season and saw which one of them they are.
Right. So the thing is like sometimes your plots they get up, and then these points were chosen based on when I took the visual observations. So the visual field observations. So I just picked those 2 dates. But if you look at the temporal like different days, you can see sometimes the plots can get up. Like it's just really maybe like this is something that can be looked at.
Unlodges or like.
Yeah.
Okay.
So I mean it can like it's not always like 100%, but you know if it's on the scale of 1 to 10, 10 being like fully lodged, it can come back to like 7 or 6 or something. So it changes like with time. So maybe yeah we can map that trade-off. See that.
Do we have any questions on the Zoom? With that, we can say thank you.
This has been a production of Cornell University, on the web at cornell.edu.